Many times when you're working with ecological communities, you may suspect that there is some grouping in your data. For example, when you have different sites or locations, or if you have different treatments in an experiment. Cluster analysis tries to group your data such that there is greater variation between clusters than within them. There's more than one way of doing this, and so there's more than one way of doing cluster analysis. Cluster analysis is used to form groupings, or clusters, from otherwise undifferentiated data. And there are two sorts of broad approaches that you can take to do so. They're called hierarchical and non-hierarchical clustering, or disjoint clustering is another name for non-hierarchical clustering. Furthermore, within hierarchical clustering, there are two more divisions. There are agglomerative approaches, which emphasize similarities in data, versus divisive approaches, which try to identify differences or distinctions in your data. The agglomerative approaches are more common in ecology, and within them there are single linkage methods, complete linkage methods, and average linkage methods. Non-hierarchical clustering, or disjoint clustering, the most common type of it is called k-means clustering. Okay, so you've got these two broad kinds of ways of doing cluster analysis, hierarchical and non-hierarchical. Each has its advantages and disadvantages. So when should you use what method? First off, you need to understand the distinctions between them. Disjoint clustering is simply a division of your data into non-overlapping clusters, such that each data object is in exactly one subset. In contrast, hierarchical clustering, if you think about what the word hierarchical means, hierarchical clustering forms a set of nested clusters that can allow you to discern some emergent patterns about your data that relate to scale. So if you're interested in potential scaling effects, then you're going to want to take a hierarchical approach. If you have some a priori ideas about the number of clusters that may exist in your data, then disjoint clustering is often preferred because you know what the value of k is going to be. But beware of confirmation bias if you do that. If you do not have such a suspicion about how many clusters you have, then hierarchical clustering is generally advised. Disjoint methods are more computationally intensive than hierarchical ones. And in disjoint clustering, you start with random cluster assignment. So the results produced by running the disjoint algorithm repeatedly many times may differ. What you're looking for is consensus. Whereas hierarchical methods are exactly repeatable. Hierarchical methods can be used on more different types of dissimilarity or distance metrics. And disjoint methods are less sensitive to outliers, and in fact they're very useful in identifying outliers. So what you need to do is you need to evaluate the type of dissimilarity or distance matrix that you can use depending on your data, whether you have some preconceived notions about clustering in your data or not, and if so, how many clusters might exist, whether you have outliers or are interested in finding outliers, and how much computer firepower you have. And that's especially important if you have a very large data set. And then you can make your decision accordingly about whether to use a hierarchical or a non-hierarchical approach. So now in the subsequent slides, I want to go through each one of these methods in more detail. In hierarchical clustering, you can group things on the basis of similarity, that's agglomerative, or by identifying distinctions, and those are divisive approaches. The hierarchical part of hierarchical cluster analysis, the hierarchical component constructs groups within groups by either of these methods, with the strongest differences among groups at higher levels. Agglomerative approaches are more common in ecology because they're more useful at identifying smaller clusters, whereas divisive approaches are better at finding large clusters, which are generally already readily apparent in ecological data. There is an R package called cluster that has two functions that allow you to do agglomerative or divisive cluster analysis. Those are Agnes for agglomerative and Diana for divisive clustering. And you can type in $Agnes and $Diana in R to pull up help and find out more details about these. We will not be using these today because these functions, it packages a lot of things together that you need to be able to understand individually. So agglomerative approaches are the ones that are generally used in ecology. And so in the next slide, I want to talk about the output that you get from an agglomerative hierarchical cluster analysis and how you interpret it. In an agglomerative hierarchical cluster analysis, the two sites that are the most similar, or least dissimilar, to each other are fused at what's called a node, forming the first cluster. This is repeated with subsequent sites fused one by one in order of highest similarity to the cluster to which they are most similar. And eventually all sites are accounted for. And so in all forms of agglomerative cluster analysis, you eventually end up with everything clustered together into a single cluster. This is displayed graphically in this kind of plot. This kind of plot is sometimes called a dendrogram or a tree, or if it's arranged vertically like this one, it's called an icicle plot. You can also see these kinds of plots arranged horizontally. You just rotate it. And in this dendrogram, each branch tip down here corresponds to one sampling object. As we move up the tree, objects that are similar to each other are combined into branches, which are then themselves fused at greater heights. On the left, you may have an axis. We don't have one for this particular dendrogram, but you may have an axis that's called height or dissimilarity. The higher the height of the fusion, the less similar the objects are. Now in an icicle plot like this, we can only gauge similarity on a vertical axis, not on the basis of proximity horizontally. We cannot use the proximity of two objects along the horizontal axis as a criteria of their similarity. That is, just because two sampling units are next to each other in a dendrogram does not mean that they're more similar to each other than they are to other sampling units that are in the same group. Okay, so this axis means nothing to an icicle plot. This axis is the one that we're looking at. So what you have here is you have all of these sites down here on the bottom, and you see these are fused here. This is a node, and then where they're fused again, there's a node, and here's a node, and here's a node, and so on and so forth. And so this axis of dissimilarity indicates where you are seeing clusters. So remember, this is hierarchical clustering. There's clusters within clusters. So here's two clusters, and then this one is further broken down into two, and this one is further broken down, and so on and so forth. Okay, so you can see how this is generated. And there are three methods for doing so, three different linkage methods. There's single linkage, complete linkage, and average linkage methods. The single linkage methods are sometimes called nearest neighbor methods because they emphasize similarity in units. The problem is that they may overestimate similarity, and you wind up with what's called chaining. So instead of having a nice sort of spread out tree like this, you get one that is more vertically arranged. And here's an example of this over here in a couple of trees that are horizontally arranged. These trees were generated with the same data. The top dendrogram was generated using an average clustering method and then average linkage method, and the one down here was generated with a single linkage method. And you can see here what I mean by this chaining here. The complete linkage method, on the other hand, is sometimes called the farthest neighbor method, and it sort of takes the opposite extreme, and it can overemphasize differences that exist. So single linkage can underemphasize differences, and complete linkage can overestimate differences. And so as a consequence, average linkage methods are the ones that are generally used, especially in ecology, followed somewhat by complete linkage methods, and then single linkage methods are generally not recommended. Okay, so now in the next slide I'm going to talk about how we actually do this cluster analysis so that we eventually get to this end result, the dendrogram. So in an agglomerative approach, in any kind of cluster analysis, you need to start with a dissimilarity or distance matrix. And so the question is, what type are you going to use? Well, that depends upon the structure of your data. And so what I want to do now is sort of go through the logic process of picking a dissimilarity metric to use. And so in the online notes, the example I used was from the grasslandcommunity.csv file. And if you look at those data, you see first off that it's a homogeneous data matrix, meaning that it consists of a single data type. It is numeric, and it's percent cover data, meaning that the numbers are between 0 and 100. And so because of those things, those traits eliminate Steinhaus and Hellinger metrics. There are lots of zeros in this data set, and so that tends to exclude Euclidean, Manhattan, and Canberra. The Mahalanobis approach really isn't needed here, and chi-square de-emphasizes abundant species, of which there are some in this data set, so that's probably not the best choice. And so therefore, sort of by default, the Bray-Curtis metric is used. And so this is the kind of logic that I use, or the steps that I use to pick what kind of dissimilarity matrix I'm going to use. Okay, so now that I've picked a distance metric, then there are a couple of different packages and a couple of different commands that you can use. So for example, vegan has the command of vegdist, whereas the basic stats in base R uses hclust. And you have different ways of doing this, and each one of these different packages has different pros and cons about it. And we worked with these in the online notes. So when we perform an average linkage cluster analysis to the grassland community data, we wind up with this dendrogram. You read a dendrogram from the top down. The vertical axis is really similarity. You can see how the bottom branches are individual units. They have a similarity of 0. In other words, they are individual units. So how would you interpret this dendrogram? Well, remember that in the grassland community data, each one of the site names included an abbreviation as to what habitat type it belonged to, mixed grass or fescue, which was really nice. And so you can see here how all of the mixed grass sites cluster together, form a cluster, and then how all of the fescue sites form a different cluster. And so what this indicates is that it looks like that mixed grass and fescue habitats contain different plant communities because they separate out into 2 clusters. And you can also see differences among sites within habitat types for both mixed grass and for fescue. So then you can use these clusters to examine patterns in the rest of your data. Like, for example, if you were interested in the effect of some environmental variable, like aspect, for example, in driving patterns in your data, then you can do something like a box plot. And the way you interpret this is the clusters here, the cluster number 1 were the mixed grass habitats, and cluster number 2, these were the fescue habitats. And so you can see here that the sites comprising cluster 1, the mixed grass sites, have a much wider range of aspects than sites that are fescue, which only occur at a few aspects. What this suggests is that fescue is more sensitive to aspect and maybe some variables that are correlated with it, like temperature or moisture or whatnot, than is mixed grass. And so these differences indicate that this clustering is an ecologically logical one. Let's go back to our original dendrogram. How can you tell whether it's a valid representation of your data? Well, what you can do is you can compute a correlation between the values on the vertical axis, the height or the dissimilarity or distance, which are more properly called cophonetic distances, hence this command here, cophonetic, the correlation between those distances and your distance matrix. In our case, the resulting correlation coefficient is very close to 1, which indicates excellent fit. But you shouldn't just use the correlation coefficient because the average linkage method generally produces a higher correlation than other agglomerative methods. So don't just look at this number. You also need to look at the tree structure to see whether or not it's logical. And there is actually a package called pvclust that allows you to statistically examine each branching on a dendrogram. It produces a p-value. It uses a bootstrapping approach to produce a p-value for each branching. And if you want a worked example of this, then you can reference this book by Gardner, 2014, Community Ecology, Analytical Methods Using R and Excel, which I have noted in the course website and referenced in the notes for today. So if you want to look more about pvclust, you can do that. Here's an example of how different clustering methods on the same data set can produce somewhat different results. And so we have two dendrograms here. The top one was produced by the average linkage method, and the bottom one was produced by the complete linkage method. And it is readily apparent from these figures that the clustering patterns differ by method. But even so, both of these methods indicate that mixed grass and fescue separate into distinct clusters. Okay, so this has been an example of hierarchical clustering. Now let's move on to non-hierarchical clustering. In non-hierarchical clustering, also called disjoint clustering, the most common method is called k-means clustering. And k is the number of clusters. You designate k. For this example, using the grassland community data, I just picked k of 5 just for illustrative purposes. But you need to choose a number that is based on some sound reason. And in a few slides, I'll discuss how you might do so. What you wind up with, the result is this kind of plot, which is called a silhouette plot. You use the function PAM in cluster, and it produces a silhouette plot here. And so here are my 5 clusters over here. And the site names are cut off here, but anyway, here's all my sites. And for each cluster, it shows the number of sites per cluster, which is the number of horizontal lines, which is also given in the right-hand column over here. The mean similarity of each site to its own cluster minus the mean similarity to the next most similar cluster, which is given by the length of the lines. with the mean also in the right-hand column here, and the average silhouette width. Sites that fit well within their cluster have a large positive silhouette width. Those that fit poorly have a small positive or even a negative silhouette width. But I have seldom seen silhouette analysis in ecology, perhaps because large positive silhouette width is a relative measure. How large is large? So anyway, this is one form of output that you can get from k-means clustering. It's more common to see output like this. In this plot here, you see the sites by name, and you see ovals here. And notice the overlap in a couple of the ovals here. This suggests that these aren't two distinct clusters, but perhaps are a single cluster, whereas these others out here are pretty distinct, and so perhaps they are valid. But remember, we had two main clusters, and you may think, well, gosh, this makes it look like there ought to be, 4, I would think. But remember, there was clustering within clustering that we saw from our hierarchical kind of analysis, which induces that sort of effect. A non-hierarchical cluster analysis can reveal differences in the patterning in your data. But again, when you look at these, you see how all of the mixed grass sites sort of cluster over here on the left-hand side, and all of the fescue ones cluster over here on the right-hand side. So again, there is evidently some real logical distinction between mixed grass and fescue sites. And just like we did earlier in the hierarchical cluster example, you can use the resulting clusters from a disjoint cluster analysis to examine patterns in the rest of your data. And so once again, looking at differences with aspect by cluster. And so again, you get a box plot. And so how would you interpret this? You can see that clusters 1 and 2 have those sites represent a wider range of aspects, whereas clusters 3, 4, and 5 represent sites that are all at a very narrow range of aspects and very mostly similar aspect values. So this indicates that possibly that clusters 1 and 2 really should form a single cluster, at least on the basis of aspect, and clusters 3, 4, and 5 form a separate one. And so this means that my initial k equals 5 may not be ecologically valid, at least for this environmental variable. In k-means clustering, you have to designate the value of k. And earlier I said that this should be predicated on something that you are examining in your data. You need to have a sound reason for the number that you designate as k. And so if there are two distinct habitat types, like mixed grass and fescue, then k equals 2 would be a logical choice. But if you don't have a clear idea of the number of clusters a priori, there is a way to help you choose a value of k to use. Go back to your distance matrix and create a hierarchical cluster object. So in our case, our distance matrix here, and we use this command to create a hierarchical cluster object that you can then cut here into different numbers of clusters. In this example, I chose to examine between 2 and 5 as the possible number of clusters present in the grassland community data. And this is the output that you get. And so what you're looking for is something that is logical and not sort of choppy. See how this goes from 1 to 2, back to 1, then 3 to 4 to 3. That's what I mean by choppy. Okay? With these data, 5 is really choppy. Whereas 2, there's ones, okay, here, and then it goes to 2, back to 1. Oh, but the mixed, yes, it goes back to mixed. Okay, so that 1 to 2 to 1, okay, that's logical. So that's what I mean by what you're looking for. So it looks like 2 is most appropriate, maybe 3, possibly. These two fescue sites here, K14, K15, and maybe K17 here differ from other fescue sites. And so by looking at the number of clusters, you can potentially identify outlier sites in your data. So it's possible that these 3 sites, fescue K14, fescue K15, and fescue K17, form a cluster of their own. So let's go back and designate 3 clusters and rerun our analysis and see what we get. And here's the dendrogram that we get from k-means clustering, where k equals 3. And if you examine the validity of this dendrogram via the correlation analyses that were mentioned earlier, you'll find that the result will be the same as for the dendrogram produced before changing the value of k. So changing the value of k does not change your correlation coefficient. And in the question in the online notes, I said, why do you think this might be so? The answer is, look at the topology of this tree. The topology of the tree doesn't change by designating different numbers of clusters. Look at this tree versus the one that's in page 9 of your online notes. It's the same one in terms of branching. So again, that correlation analysis you should not use it as the sole means to say, yes, this is how these data cluster out. No, you need to use multiple logical means. So in this case, what I would do is I would look at this and I would say, okay, this does look like there is some difference, some distinct difference in these 3 fescue sites here. They are certainly different from these mixed grass sites. And of course, at a higher level, of course they're going to be grouped with the other fescue sites because they're fescue. But they are probably distinct enough to be considered a distinct cluster of their own. Up to now, we've worked with abundance data. The grasslandcommunity.csv had abundance data in the form of percent cover values, for example. But sometimes all you have is presence-absence data. You can perform a cluster analysis on such data not in terms of distance, in terms of beta diversity. If you need to refresh your memory on how beta diversity quantifies similarity across sites, then go back and read the diversity indices notes. So GB BIOL is a presence-absence data set, and you can use the command beta diver in vegan to get your beta diversity values, as opposed to Bray Curtis and so forth. That's what we're going to be using here. And then we can use hclust to perform a hierarchical cluster analysis. And here is the resulting dendrogram. And from this dendrogram, you can see that sites from each of the 3 habitat types form distinct clusters. Wood sites are the most distinct, whereas grass and edge are relatively more similar to each other than either is to wood, at least in terms of beta diversity. And if you look back at the notes from the lesson on diversity indices, you'll see that this result is consistent with other analyses you did that compared these habitat types. Okay, so now I want to cover a few empirical examples from the ecological literature. In this first study, these researchers used the Bray-Curtis dissimilarity metric in a hierarchical cluster analysis to identify 5 distinct assemblages of reef fish out of 266 species that co-occur on coral reefs in Vietnam, with groupings based on various environmental variables such as coral type and water depth. And this is what they got here. However, these folks did not specify what type of hierarchical clustering method they used, and nor did they evaluate whether their tree was valid. So this is an example, but this is one that is lacking something that really should be added whenever you do a cluster analysis so that people can have an assessment as to whether they should trust your results or not. And here's an example of a non-hierarchical cluster analysis that was done where you see that there's an actual silhouette plot. This is one of the very few examples from ecology that I could find. These folks used cluster analysis to detect the presence of a relatively homogeneous homogeneous groupings of calls in wolves. The silhouette plot was used as a method of cluster interpretation and validation. And this let them determine that there were 6 call types in gray wolves. For your assignment, we're going to be using our old friends, the ground beetles, and you're going to be using ground beetle abundance data. So you are tasked with performing a cluster analysis on these data. You're going to use a Euclidean dissimilarity metric and then perform a hierarchical cluster analysis on it using an agglomerative approach with the complete linkage method. And in the online notes, I talk about how you should relativize data before you do a cluster analysis, but you can skip it for this step, for this question. And then you're going to produce a dendrogram, you're going to verify it, and then I want you to interpret what it means. As a hint, in the notes, there was an area where I had used a hang function. Don't use that because the hang function pulls everything down to a similar level at the bottom of your dendrogram. Just let everything hang naturally, and that will allow you to potentially see anything that's odd about that dendrogram. And if there is something odd, then what you can do is perform a k-means cluster analysis to identify, plot, and label meaningful clusters. And now, then after you do that, you're going to use those same data to compare the complete linkage method to single linkage and average linkage methods. And you want to see whether these 3 methods, whether they converge, and if not, then which seem to best represent the raw data.